And so we were very pleased to have Nina Bhardwaj agree to come and speak about her work in immunotherapy of cancer and telling us a little bit about what it takes to get things out of the lab and into patients. Nina.
Well, thank you very much, Daryl. It's a pleasure to be here. And I'm actually going to talk about a new story. Since this was an engineering symposium, I thought I'd bring in some new concepts that might hopefully provoke some discussion. And so what I'm going to talk to you about today is about basically how silent pericentromeric repeats that are expressed in cancer cells and often during immune activation can actually engineer immune response on the part of a dendritic cell, and how this might be taken advantage of in order to induce immunity in patients with cancer. So I want to acknowledge a number of people that I've worked with over the years. This is sort of a story that's been progressing over the last 5 or 6 years, largely through work that I was doing with Ben Greenbaum and Arnie Levine when Ben was still at the Institute for Advanced Sciences in Princeton, and with postdoc Sonia Jimenez Brenda. And at that time, we were working on actually influenza virus genomes and evolution of influenza virus and activation of dendritic cells. And that story, which I'll talk to you about briefly, led us to a whole new direction to look at essentially non-coding RNAs in cancer. And that work was done by a wonderful postdoc in my lab, Antoine Tan, who's now at Agenus, the graduate student Luciana Muniz, and our colleague David Ting at the MGH, and also Andrei Gudkov at Roswell Park Cancer Institute. So essentially, this is the message of the talk, and that is that silent pericentromeric repeats, these are repeats that are present in the pericentromeric region that normally are not transcribed, but can be in cancer cells, can engineer immunity. And what I'll start by showing you is that from our work with influenza virus is that the evolution of RNA viral genomes can be shaped by pressures that are observed in the host genome. Subsequently, the pressures on these viral genomes have helped us inform motif patterns in host non-coding RNAs. And finally, that non-coding genomes, which are apparently expressed in cancer cells, can activate the innate immune response. So it's a journey starting with RNA viruses, understanding how they evolve, and learning from their motif changes through evolution, and in concert with the host genome, can actually inform us about motif usage in non-coding RNAs that are usually suppressed. So the story starts with the 1918 influenza virus, and as you may remember, this was a terrible and a very, very pathologic virus resulting in over 50 million deaths worldwide. Recently, a group led by Kawakawa showed that if the virus was used to infect monkeys, this could actually replicate what happened in humans over close to a century ago, resulting in tremendous pathology, very rapid death, inability of these animals to really mount an adaptive immune response, not sufficient time, terrible cytokine release kind of syndrome. And they hypothesized that a dysregulated immune response was really responsible for the untimely death of these animals. So just to remind you, the innate immune responses to influenza virus are multifactorial. Probably the most important one is TLR7. The virus will enter a dendritic cell, for example, and in the endosome, its single-strand RNA will trigger TLR7. In TLR7 deficient mice that are MX1 sufficient, these animals fail to clear a highly pathogenic virus. But there are also other pathways that are involved. So TLR3, which recognizes double-stranded RNA in infected cells. Intracellularly, the virus, once it begins replicating RNA synthesized, can trigger the NLRP3 inflammasome and also the RIG-I MAVS-dependent pathway that can also lead to a type 1 interferon response. So the question that Ben and Arnie and I asked is to try to understand as the influenza virus evolves, what are the pathways used to activate innate immune response, because we felt that was dysregulated in this early virus. And then secondly, once the virus, which in case of the 1918 virus is an H1N1 virus, because it's been circulating and evolving over time, it's not as pathogenic anymore. What can we learn from motif usage that might inform us about how innate immunity is activated? And so Ben, who is the systems biologist here and the mathematician and the physics person, has been teaching me a lot over the years in my old age about how to analyze these changes, showed the following. So if you look at the H1N1 1918 virus here, what he found most importantly was that the frequency of the CpG dye-motifs in the virus over time as it's circulating in humans declined substantially. This is in contrast to more recent viruses like the avian flu viruses and H5N1, highly pathogenic virus, which still retains a lot of CpG motifs in their RNA. Then you have the reassortant viruses down here that have been also circulating in humans, which are gradually also showing a decline in this CpG motif. And then I went on to using algorithms that were developed to look at the frequency of the CpG motifs relative to the total CG content in various human RNA positive and negative strand RNA viruses. And you can see that this odds ratio is far below 1, meaning that there is some sort of force or entropy that is reducing the number of CpG motifs. And most interestingly, if you look at the human genome, 22,000 genes or whatever, and you look at the CpG motif in our genomes, CpG is also highly underrepresented in our genomes. And if you map these RNA viruses to our genomes, you can see that over time, these circulating RNA viruses come to look a lot like our own human genomes. And what we went on to show was that CpG and UPA actually motifs drop over time, whereas CPA and UPG go up. And interestingly, a lot of cytokines involved in innate immune response or adaptive immune response, like type 1 interferons and IL-2, either have an extremely low or no CpG content. So we, as well as circulating RNA viruses like flu over time, have learned to prevent actually an autoimmune response as such, if you want to think of it that way, by actually reducing the number of CpG and associated UPA motifs in our genomes. So it's not just simply CpG. If you look at the bind H1N1 sequences, you'll see that CpG in the context of surrounding Cs or Gs are actually protected. As shown here, this frequency doesn't change in the H1N1 virus. But when it's in a U or an A context, the frequencies do go down. And if you look at the 1918 sequence, you'll see that the most overrepresented CpG sequences, as shown here, shown you the four most overrepresented, are usually in the context of the uracils and the adenines around it. And these will drop dramatically. So if you look at current viruses, you'll have relatively few. So going back to the 1918 virus, the question we asked was, are these sequences then important for inducing an innate immune response at all? And so what we did was to synthesize these RNA sequences, and with DOTAP, transfect plasmacytoid dendritic cells. And as you know, plasmacytoid dendritic cells, in this case, these were human cells, are the most important type 1 interferon producers in the body, produce the most type 1 interferon. And they also express TLR7. So what you'll see here is if you look at the sequences V1 through V4, as shown here, the ability to induce type 1 interferon goes down dramatically as the number of uracils around the CpG sequence drops. If you switch the CG to GC, it's in here, you also reduce the ability to make type 1 interferon. And if you eliminate all the uracils and replace them with adenine, you also deplete the type 1 interferon response. And this correlated also with TNF production at the PDC level and NK cell activation. So this was very consistent with the fact that these sequences could in fact induce a potent immune response at the level of the PDC. So what I've shown you so far is that really the number and placement of the uracils around the core CpG motif are critical. The motif itself, which can completely control interferon alpha production, even when the U content is lowered. And we showed in those experiments that we could block this induction of type 1 interferon by using a TLR7 inhibitory ORN sequence. So essentially, these sequences were triggering PDCs via TLR7. The question we asked was whether these motifs were really critical for the virus to induce innate immune activation. And so we constructed an H1N1 virus, it was shown here, WSN, in which one of the polymerase genes was replaced with a GFP cassette, two GFP cassettes. In one case, a cassette that contained a low number of the immunostimulatory CpG motifs, and in another case, it was replaced with a cassette containing a high number of the immunostimulatory CpG motifs, and ensured that the protein sequence was unchanged. So these viruses were then used to infect PDCs. And what we found was that the low CpG GFP virus induced far less type 1 interferon than the high GFP, the high CpG GFP containing virus. And of course, since we're only replacing one gene, we wouldn't have expected all the type 1 interferon production to be lost in the low CpG containing virus. So these studies were done in vitro, but since then a number of other investigators have actually shown similar data in either in animal models, and in particular, for example, a point at work from Burns, who showed that dinucleotide composition of viral genomes affects host pathogen interactions for poliovirus. This is also true for SIV and HIV, where a biased nucleotide composition has been shown to be related to pathogenic potential, and more recently work showing that CpG frequencies in influenza can also affect pathogenicity and the response to the virus. So lessons that we learned here were that certain motifs have a pattern of avoidance in the host, namely the CpG motif, in the context of uracils and adenines, and that viruses that are circulating in the host evolve towards really mimicking host motif usage. And the immunogenic viruses are those that are going to have a very strong bias in motif usage patterns. And so this made us think about, well, if the host genome has suppressed these CpGs motifs over time, what about those genes that are not usually expressed in the host genome, but which are expressed under aberrant conditions, such as an infection or in cancer? Are those motifs going to be subjected or not to the same forces? And if they're not, one would predict then that they would actually function as immunogenic, danger-associated molecular patterns, and as such could be taken advantage of to stimulate the immune response in cancer. So what are those non-coding RNAs? They've been referred to as dark matter genome. It's comprised of many repeated sequences, as shown here. In fact, our genome, only 2% of our genome encodes protein, and the rest of it is made up of these repetitive elements and other genes. And now that the human transcriptome is being better and better analyzed and annotated, we now know that in the human transcriptome, about a third of our transcriptome comes from non-coding RNAs, which has been really a big surprise. So there are a number of different repetitive elements that comprise these non-coding RNAs. They include tandem satellite repeats that are in the pericentromeric region. Then there are the signs and lines, which are short interspersed elements. throughout the genome. There are also the DNA transposons and the LTR transposons as well. And altogether, there are about 5 million copies of these in our genome. So what do non-coding RNAs do? They're important regulators of gene expression in many different ways. They can, through interactions with chromatin, with proteins and so forth, modulate transcription. They can interact with transcription factors. They can target mRNA splicing, stability, translation. They can maintain, the satellite repeats, for example, can maintain heterochromatin architecture, enabling the kinetochore formation to be sustained and driving chromosomal segregation through mitosis to allow duplication of the genome. So these are the functions, and we're still learning more and more about the functions of non-coding RNAs in the steady state or under so-called normal conditions. But what happens in cancer when a lot of non-coding RNAs that aren't really expressed during sort of the day-to-day situation are expressed? What happens in those circumstances? And I think one of the first clues that these non-coding RNAs that are aberrantly expressed in cancer cells came from work by our colleague Andrei Gudkov. And he was studying cell lines that were deficient in p53, as shown here. And when he looked at the transcriptome of those cells, what he found was upregulation of a number of these non-coding RNAs. When these cell lines were then treated also with azacitidine, a demethylating agent, he was able to show a massive upregulation of and expression of these non-coding RNAs, including endogenous retroviruses, in particular one pericentromeric repeat called GSAT, and that's shown here. He also found a number of signs and other non-coding RNAs as well. But when he looked closely what was happening to the cells, these p53 deficient cells that were treated with azacitidine, he went on to show that they express a lot of type 1 interferon beta 1, a lot of ISGs, OAS, MX1 and 2, IRF7. And most dramatically, that these cells, when treated with azacitidine, died of apoptosis. And he called this a transcription of repeat-dependent activated interferon response, or a TRAIN, and went on to suggest that the loss of p53, together with the loss of DNA methylation, led to the expression of non-coding RNAs that are not usually expressed, and that in turn induced a type 1 interferon response, leading to apoptosis. So normally, p53, DNA methylation, epigenetic suppression of these non-coding RNAs will prevent their expression and keep the cells from dying. So this is also true in vivo. We showed that in various tumors, such as testicular teratomas and liposarcomas, where p53 is lacking, there is also upregulation of this pericentromeric repeat GSAT, as shown here and here. And in p53 null mice and thymic lymphomas, you'll see, again, GSAT expression in both directions, actually, both sense and antisense. And this is correlated with upregulation of interferon beta 1 in vivo as well. So what I've shown you so far is that expression of these satellite repeats induces a type 1 interferon response that seems to be correlated with expression of various, not just repeats, but also various other non-coding RNAs that are normally suppressed. So the obvious question was to ask whether these satellite repeats are also upregulated in human cancers, not just murine cancers. If they are upregulated, do they activate an immune response? And how do they activate the immune response? Because they would not be subject to the same forces of suppression that a normally expressed RNA would be, would their motif usage actually dictate the immune response? And so those are the questions we've been asking and working on, and that's what I'll share with you next. So, in fact, these satellite repeats are overexpressed in cancers, in particular in epithelial cancers. And our colleague David Ting published a few years ago that in pancreatic cancer, up to 10% of the transcriptome comes from these satellite repeats, most notably HSAT2, which is also a short a repeat around 180 base pairs or so. He went on to show by FISH that you can actually find the expression of these repeats in very early stages of pancreatic cancer, but also in metastatic cancer and also in circulating tumor cells. So the expression of this repeat appears to be, as such, a biomarker of evidence of this cancer. It's, as I mentioned, not only expressed, overexpressed in pancreatic cancers, but also a number of other epithelial cancers, lung, kidney, ovarian, and prostate cancer. So the next question was really to ask, what is the motif usage in these repeats, since they're normally suppressed? So Ben actually went to the GenCode database to look at the non-coding RNA transcripts from just an array of cells and tissues in humans and in mice. And this is the frequency of various dinucleotide motifs based upon all this data. And what you can see is that the CpG and the UPA dinucleotide motifs are really underrepresented in GenCode. If you then compare the frequency of these motifs in human GenCode and mouse GenCode and compare them with human pancreatic cancer or mouse cancer cell lines, you'll see that in these cases, the CpG motifs are actually overrepresentative, just sort of what we were predicting to be the case. And then Ben did quite an amazing analysis, actually, looking at the strength of CpG statistical bias, in other words, measuring the force against which CpG is expressed or not expressed, in gray, either from all the transcripts in the GenCode database versus in pancreatic cancer. What you call in red are sort of the outliers, the ones that haven't been subjected to these forces as much. And you can see that HSAT2 is an outlier. And in murine cancer cell lines, you can see that GSAT is an outlier as well. In fact, when you compare the forces on this dinucleotide motif in GSAT and HSAT, and you compare it with things like E. coli, you'll see, which of course has a lot of CpG, you'll see that they sort of aggregate together in terms of the force exerted on CpG, meaning that the force is much less on these than it is on, let's say, a circulating influenza virus that's been in the human population for several decades. So basically, these data showed that the motif usage in HSAT and GSAT clusters with foreign RNA, and more importantly, that the force that is exerted on CpG or not exerted on CpG tends to be particularly strong when an A or U is next to the core motif. And this is, remember what I showed you for influenza virus as well. And these are just the outliers, just to give you a list of what we identified as being outliers in the non-coding RNA genome. And I just want to point out that HSAT, for example, is a relatively new insertion into the human genome. It just became evident in primates, but others tend to go back much further. These also encode other signs, lines, and endogenous retrovirus sequences. So we then went on to look more carefully at the sequence in HSAT that is expressed in pancreatic and epithelial cancers. And the HSAT core is about 180 bases long. And what we found is that the core sequence, the core element expressed about 14 times in HSAT, is actually the same sequence that we found in the V1 fragment that I showed you earlier from influenza virus that is highly immunostimulatory for plasmacytoid dendritic cells. And so we asked whether HSAT itself could be immunostimulatory for dendritic cells. So we created mini genes, which we call immunogenic non-coding RNA, that encoded HSAT and the mouse GSAT, We also had controls where we scramble these, as shown here, or we had endogenous non-coding RNAs that are normally expressed, not in the cancer setting, but normally expressed of comparable length. And then we took monocyte-derived dendritic cells and transfected them with these various non-coding RNAs or their scrambled versions. We also used immortalized murine bone marrow macrophages and then asked whether they could induce an inflammatory response. So here's the data showing you the effect on human monocyte-derived DCs. And as you can see, HSAT2 and GSAT induced IL-12 and TNF-alpha, and also IL-6, not shown here. And in the case of murine immortalized bone marrow macrophages, it turns out that only the GSAT is stimulatory, but you get TNF, MCP1, and IL-6 induced as well. So in fact, as we might have predicted, HSAT and GSAT are immunostimulatory for these cells. We then went on to try to identify the pathways that were being used by HSAT and GSAT in these innate immune cells. We looked at the endosomal toll-like receptors. We looked at intracellular sensors, such as the STING and the inflammasome, MDA5 RIG-I, MAVS-dependent pathways. And to do that, we used immortalized murine macrophages that had been knocked out for various toll-like receptors and sensors. And actually, most of the results showed that none of these were involved. So I'll show you that very quickly. TLR3, 7, and 9 knockout, no deficiency in GSAT induction of TNF-alpha. In the STING, NLRP3, ICE, or MAVS knockout, again, no reduction. And finally, in a number of interferon-related signaling cascades, we found no reduction in GSAT stimulation of TNF. What we did find, though, is in the MIghty88 knockout macrophages, and in macrophages that have a mutation UNC93B, which is required for transport of endosomal TLRs from the ER into the endosome, we lost the ability of GSAT to stimulate TNF-alpha in our murine macrophages. And we had something very similar using HSAT in a THP1 reporter cell line. So essentially what we showed was that GSAT requires, and presumably HSAT requires UNC93B and MIghty88 to trigger an immune response in innate immune cells. We don't think TLR8 is involved since we are working with single-stranded RNA, but we've got to conclusively rule that out. We've shown that TLR13, which is a murine TLR that happens to recognize a bacterial ribosomal RNA that actually has a sequence that's very similar to GSAT's, is not involved for GSAT recognition either. And we're now looking to see whether perhaps a complement of endosomal TLRs may be necessary for activation. But it's quite possible other intracellular sensors are required, and this is a project we're still working on. So what I've shown you so far is that HSAT and GSAT are preferentially expressed in tumor cells. They can stimulate dendritic cells and macrophages to produce inflammatory cytokines. But the question really is, in vivo, what are these pericentromeric satellite repeats doing? Are they also promoting tumor growth and/or priming the immune system? Are they having a tumor intrinsic effect, as Gukov described in the murine system? And so I'll just show you recent work from David Ting's group showing that as tumors grow, in this case the colon cancers, they actually increase in copy number expression of HSAT, as shown here. And that if you look in the TCGA database, patients with colon cancer, about 46 patients, and you look at the HSAT numbers, copy numbers, patients that have HSAT gain compared to germline do more poorly than those that don't have this appreciable gain. This is also true for other repeats that are associated with HSAT expression. And David also went on to look at tumor growth, and what he found was that HSAT copy number gain occurs only when these tumors, these colon cancer tumors, are grown in spheroids or in vivo. However, if you treat these tumors with locked nucleic acids that target HSAT2, you can get a reduction in tumor growth. And interestingly, and this is probably an interesting point of discussion, is if you treat these tumors in xenograft models with a reverse transcriptase inhibitor, such as DDC, you can also reduce tumor growth substantially, and you can reduce the copy number gain of HSAT2, suggesting that HSAT2 gain is important in tumor growth, and that HSAT2 gain is going to require some element of reverse transcription to form new copies that can then be reinserted back into the genome. So we have two situations. We have HSAT2 being expressed in cancer cells, presumably promoting tumor growth. At the same time, I've also shown you that HSAT2 can also activate dendritic cells and macrophages. So what's really going on in the tumor setting? And to try to answer that question, this work done by Antoine Tan, he's been looking at the possibility that in these tumors, as they're growing, they're generating exosomes, and that the exosomes are incorporating HSAT2, which can be released into the tumor microenvironment and can possibly activate surrounding innate immune cells. And what he's shown is that RNA taken from cancer cell lines, colon cancer cell lines that are grown in spheroids, which when transduced into dendritic cells, can in fact induce an inflammatory response in vitro. So altogether, what we're thinking now is, and again, this is work in progress, is that in aberrant dysregulation of HSAT2 as cancers grow, that RNA can, when it's expressed, because it hasn't been subjected to the same forces as the rest of the genome, expresses a lot of CpG in a UA context, could actually induce a type 1 interferon response that is tumor cell intrinsic. In addition, it could be released out into the tumor microenvironment in exosomes or other formats that could trigger innate immunity in the surrounding microenvironment, and at the same time, through reverse transcription, can be reinserted back into the pericentromeric area and also promote tumor growth. So there's sort of a constant battle in the tumor microenvironment. And I'll just show you, and as I end, one referred to another set of studies that published that seem to suggest that this is not an unreasonable hypothesis. This is work from Wang et al. Showing that TIRA, which is the telomeric repeat-containing RNA, is also dysregulated in cancer, and that TIRA non-coding RNA can actually be identified from tumor cells that upregulate TIRA in exosomes, and these exosomes can also activate PBMCs in this case to produce an inflammatory response, much like we've seen in our dendritic cells and macrophages. And you can also do this by expressing TIRA and liposomes. So just to end then, what I hope I've shown you is that a set of repetitive non-coding RNAs are preferentially expressed in cancers. We think these are normally suppressed as a result of epigenetics. They have motif usage that are very unusual, and I didn't get into this, but because these non-coding RNAs can be transcribed in the sense and antisense direction, they can form double-stranded RNAs, which may also be immunogenic. I've shown you they can engage signaling pathways downstream of pattern recognition receptors, and we're working on really how to take advantage of expression of these repeats in order to modulate the immune system in vivo. And in fact, what we're doing now, and hope to talk to Daryl more about this later, is we're actually injecting these repeats, which we synthesize directly into tumors, also using them as adjuvants in vivo, in the hope that these will really be very, very strong adjuvants for priming anti-tumor responses eventually in people. So our study just adds to a host of other studies, and because of time I won't go into them, that are now indicating that RNA and DNA derived from tumors, whether it's a result of azacitidine, radiation, chemotherapy, has the ability to be released in the tumor microenvironment and initiate an anti-tumor immune response. So I've thanked most of the critical people who worked on this. It's been really, I hope you got a sense of the journey, which started a number of years ago, but is now, I think, giving us a lot to do in cancer, and really thank a number of other people who provided us with the cell lines that I mentioned. So thank you very much. Stop there.